I like to use pictures to illustrate stories. I don't like to use bulleted text as much. So here is my picture that I'm going to use to help us tell this story of the reuptake of neurotransmitter. Right? We've just told the story of the release of neurotransmitter. I'm not showing snares and stuff. We've fused and we've released neurotransmitter. And it is interacted with receptors, and stuff has happened in the postsynaptic cell. That's all happened. Now we have to end this process by taking this neurotransmitter up and putting it back into the vesicles where it came from, where we were for the next release time. So we've got membranes in black, we've got neurotransmitters in black, we've got receptors in black, but we got some other things, proteins, in color. So what's the first? Let's talk about this. It doesn't matter what the color code is. In fact, it doesn't even matter what we go first. Let's label this picture and then tell the story. So somebody tell me the name of a protein that is going to be important in this process, and I will put it on the board where I think it belongs. All you have to do is tell me the name of a protein that is important in the process of reuptake and repackaging of neurotransmitter. Sodium pump. The sodium potassium ATP ACE. The sodium pump. Creating the sodium gradient. Low sodium on the inside, high sodium on the outside. Okay? Yes, that is a correct answer. Another group. Henry.
The sodium-calcium exchange pump.
The sodium-calcium exchanger is there. It's important for calcium homeostasis, but it is not key to this story. Olivia.
A proton pump?
A proton pump. Okay, good. A proton pump. Again, hydrolyzing ATP pump protons. And so it's here and here. So it's in all of these vesicles. Is the proton pump. A vesicular proton pump. Making a proton gradient. Okay, next. Jenna.
Proton neurotransmitter exchanger.
A what?
The proton and neurotransmitter exchanger.
A proton neurotransmitter exchanger. In red. A proton, also, red is the protein. A proton neurotransmitter exchanger. Protons going down their gradient, neurotransmitters being pumped from cytosol into the vesicles. A proton neurotransmitter exchanger. What color is my proton? Orange. Orange blue. Okay, so there's one more. What's that one? Zoe.
The sodium neurotransmitter pump?
A sodium neurotransmitter co-transporter. A sodium neurotransmitter. No, sodium should be in blue. I'm going to try to be obsessive about this. A sodium. My neurotransmitters are black. neurotransmitter cotransport. It's here, but okay, let's put them where they belong. They belong here, and here, and here, and here. Sodium, in blue, sodium neurotransmitter. Sodium neurotransmitter. Reuptake. Here's the story. We just, by telling, so let's go through it in sequence. The sodium pumps creates a sodium gradient and maintains the sodium gradient, even as these transporters are allowing sodium to flow in, and even as voltage-gated sodium channels are allowing sodium to flow in, these pumps are pumping it back out, maintaining this gradient. This gradient is used by a cotransporter using the energy of the sodium gradient to pump neurotransmitter into the cytoplasm. At the same time, a proton ATPase is maintaining a proton gradient of these vesicles. That proton gradient, the energy in that proton gradient is used by an exchanger to allow protons to flow out down their concentration gradient and pump neurotransmitter back into vesicles. That's a story you want to be able to tell. That's physiology, so it's not on the quiz. Let me just take this opportunity to remind you. The quiz is due today, 9 p.m. today. You pick it up at the CST desk, sit down and take it, turn it in. It's a closed book. Leave your stuff at home to avoid the temptation to look something up. Closed book. If you look something up, you're cheating. Don't do it. Remember your academic integrity. If you have your phone in your pocket, it is just so easy to say, oh, I know that. I'm blanking on the name. Let me just look it up. I know what it is. I'm blanking on the name, but I know it. Well, blanking on the name and knowing it are sort of mutually exclusive. So if you leave your phone at home, you will be not tempted to look it up, because that's cheating. Okay? The quiz is anatomy. This is physiology. I can guarantee something like this will be on the exam, so now I'm taking the opportunity to remind you the exam is a week from today. And the exam will have both anatomy and physiology. Okay, so reuptake. Your handout should look something like this or have the words that we use to describe this process. I want to talk about an important concept now before we transition into details about neurotransmitters and receptors. So here's a presynaptic terminal. Here is a mitochondrion making the ATP necessary to pump, for the ATPases that we just introduced. Here are vesicles releasing neurotransmitters, these little triangular things. There's neurotransmitter release. Neurotransmitter reuptake is not cartoon. It's cartoon. What we're talking about here is modulation of neurotransmitter release. Neurotransmitter release is not all or none. It's a graded problem. The action potential is all or none, but neurotransmitter release is not all or none. We don't release all the neurotransmitter, we release some of it. And it's graded by modulatory processes. I want to introduce two types of modulation. presynaptic modulation and autoreceptor modulation. So this concept of presynaptic, the jargon is a little confusing. This is the presynaptic terminal of the synapse we're talking about. And this is the postsynaptic dendrite of the cell that we're talking about. But there's thousands of other axons going on, neurons packed into this area. And there's such a thing as a presynaptic modulatory neuron, interneurons typically. This neuron is making a synapse on this neuron. So the pre and post here sort of breaks down. This is pre to this synapse, so pre-post of this synapse. Pre-post of this synapse. Well, this is the main synapse that we're talking about, and this is the modulatory synapse. So I'm going to try to, this is still going to be called the presynaptic terminal. And this is the modulatory terminal, something like that. So now, there are neurons that make their synapses on other neurons' presynaptic terminal. So this is now introducing a new concept that not all neurons make their synapses on dendrites. Some neurons make synapses onto synapses, or even onto axons. Okay? So that's an interesting high-level information that you're getting. There are receptors in the postsynaptic cell, but it's interesting. There are receptors on the presynaptic cell to the neurotransmitter that it released. And that's why they're called autoreceptors. They receive its own signal. These signals, either from a presynaptic modulatory neuron or from the autoreceptor, can be positive, causing facilitation, or negative, causing inhibition. So now I want to take another 5-minute interval, and I want you to answer these questions. The presynaptic receptor at this synapse is a nicotinic acetylcholine receptor. You worked with this in your problem set. I'm trying to reinforce some information from your problem set. You have to remember what a nicotinic acetylcholine receptor does. So this neurotransmitter here is acetylcholine, and it's interacting with a nicotinic acetylcholine receptor. Does that facilitate or inhibit the release of this neurotransmitter? Let's just say it's glutamate. So this is acetylcholine, nicotinic receptors, some other neurotransmitter, doesn't matter. Could be GABA, could be glutamate, could be whatever. If this neuron fires action potentials and releases its neurotransmitter, acetylcholine, is that going to inhibit or facilitate the release of neurotransmitter here? And also, let's just do these two, I'm going to set up the questions. This receptor is a GPCR, G protein coupled receptor, that is coupled to GI, which is GI is a G protein that is coupled to potassium channels. So activation of this receptor and the GPCR story that you're now trying to pack into your brain, right? You know, conformational change, GTP, GDP dissociation, GTP association, leads to activation of potassium channels here. Does that cause facilitation or inhibition of neurotransmitter release? Work on that for a few minutes, and then we'll reveal.